Welcome to the modern pharmaceutical lab. Under strict GMP and ICH guidelines, this environment operates with a zero-tolerance policy for uncertainty, especially when it comes to detecting trace impurities. Every single compound in a sample must be identified and quantified with absolute precision. If an active ingredient degrades or a toxic solvent goes unnoticed, patient safety is immediately compromised. To guarantee that safety, technicians rely on two cornerstone mass spectrometry topologies, LC-MS and GC-MS. But routing a sample to the wrong machine has immediate consequences. You risk destroying the sample through thermal degradation, pulling inaccurate data, and burning through expensive laboratory time. The decision between the two is never about which instrument is universally superior. It comes down to matching the machine's mechanics to the specific chemical properties of your analyte. Before a sample ever touches the laboratory sample tray, you must carefully assess its volatility and thermal stability. Those two factors dictate exactly which workflow you must use. Let's look at liquid chromatography mass spectrometry, or LC-MS. This is the required pathway if your compounds are non-volatile, highly polar, or thermally fragile. The process starts by injecting the sample into a liquid mobile phase. Because this happens at mild temperatures, heat-sensitive molecules remain completely safe. As the liquid pushes through the column, the compounds separate based purely on their polarity and how they interact with the column's stationary phase. This illustrates electrospray ionization. The liquid droplet enters the chamber and converts into a fine spray, imparting an electrical charge without breaking molecules apart. Because this soft technique is gentle, fragile biomolecules like peptide chains survive intact, allowing exact molecular weight readings. Labs rely on this gentle workflow for intricate tasks like metabolite profiling in biological samples or quantifying heavy active pharmaceutical ingredients. If you are analyzing large biological molecules that would burn up under high heat, LC-MS is the definitive tool for the job. Conversely, gas chromatography mass spectrometry, or GC-MS, is built for a completely different chemical profile. It's strictly reserved for highly volatile, extremely thermally stable compounds. Here, the sample is injected and immediately vaporized. It's then swept up by an inert carrier gas, usually helium, to begin its journey. As shown here, the vaporized sample pushes through a tightly coiled capillary column inside a glowing oven, reaching a blistering 300 degrees Celsius. As heat ramps up, compounds separate rapidly based on boiling points. Molecules with lower boiling points exit first. Once separated, the molecules hit the electron ionization chamber. Unlike the soft spray of LC-MS, this is a hard ionization method that blasts incoming molecules with electrons, intentionally fracturing them. While this shatters the original parent molecule, it produces a highly reproducible fragmentation pattern. That specific pattern acts like a chemical fingerprint, allowing a definitive match against a database. This extreme precision makes it the standard for detecting volatile organic impurities and ensuring residual solvents meet the strict limits of ICH Q3C guidelines. GC-MS provides unparalleled analytical resolution for volatile mixtures, provided your sample is robust enough to survive the intense vaporization process. When a new sample matrix arrives at the lab bench, technicians need a clear framework to decide between these two operational pathways. This matrix outlines the dividing line. LC-MS handles non-volatile, polar compounds with mild fluid dynamics and electrospray ionization to keep biological molecules whole. Conversely, GC-MS takes volatile, stable compounds into a heated oven, using electron ionization to fracture them and track down residual solvents. The rule is absolute. A fragile, polar sample dictates an LC-MS workflow, and a volatile, stable sample dictates a GC-MS workflow. This chart maps the final decision between the two machines by plotting sensitivity against resolution. By leveraging LC-MS, you achieve massive gains in sensitivity. That makes it the optimal path for safely identifying large biomolecules and complex metabolites. On the other axis, leveraging GC-MS yields maximum analytical resolution. It is the optimal tool for untangling complex volatile mixtures and pinpointing exact solvent profiles. Neither machine can replace the other. They are both essential, functioning as the twin pillars of modern pharmaceutical quality control. By carefully matching your analytical instrument to your compound's thermal stability, You ensure accurate product purity data and reliable detection of trace impurities. Mastering this alignment allows a lab to maintain strict GMP compliance and ultimately protects the health of the patients relying on those medications. To continue mastering analytical lab techniques and regulatory guidelines, subscribe to our Pharma training channel.